Hi Cherry Stems! It's been a long time, but if you don't know me, my name is Cherry May. I'm a non-binary drag artist, dancer, and makeup enthusiast. It's been like a month since I've even filmed anything. We're in a new space. I'm in my second bedroom because we're fancy. I really want to start getting back into YouTube because I just started to kind of find my footing, and then I moved and I had a million bookings and I started a new job. So I'm excited to get back into it. We're gonna get a little sciencey and we're gonna get a little stupid today. I'm gonna be going over a scientific study that I found super interesting. I'm sure some of you will too. This is a more psychology study, so they're looking into how music affects depressive and anxiety behaviors in mice. If you want to learn some psychology, some neuroscience, some biochemistry, and you want to watch some makeup in the process, keep on watching. Now that I look like a Maltese, we can get started. I'm using my lightest product today, which is this Rare Beauty foundation, which I never use in drag. I only ever use it as a concealer out of drag. One, because it has a basic concealer brush, and two, it's just, I bought it for myself. I didn't buy it for Cherry. Sorry, team. But it is really light and the coverage is not bad. So I figured I'm gonna use it. I have a lot of thoughts in my brain at this moment, so I'm gonna try and get them out. We have a few things to do before I tell you about the study and what I'm doing makeup wise. I want a almost white base. I know that that's not something I'm going to like fully achieve. I still want to look hot. I want to look cute, but I'm also gonna look ugly, and it's okay. It's literally Halloween, by the way. That's why I'm wearing my little Trixie and Katya shirt, isn't it? Can you see it? I apologize for the clownery. What was I even saying? Oh, that it's Halloween. Who cares? I just spent like 10 minutes trying to mix up this perfect Remy from Ratatoullie color, because that's what we're doing today. I know he's not a mouse. I know he's a rat. It's in the name. But he's cute. The mice that are used in research are just a common house mouse, and they're usually all white with the red eyes, or they're brown, and it's pretty boring. So I wanted to do something fun. I'm gonna use this tiny, super skinny brush to do hair-like strokes all in my contour areas and just like around the perimeter of my face. Before we get into the actual study, I wanted to tell you guys about some of my credentials. Basically, I don't want to just spew information at you. Since it looks at the effect of music on depression and anxiety symptoms, I have clinical severe depression, or MDD, major depressive disorder. I've been diagnosed with general anxiety. I've been diagnosed with OCD. So I know my way around a therapist's office, a psychiatrist's office. I have a bachelor's in science in biochemistry and molecular biology. And I know it's a mouthful, and so I usually only just say biochemistry because it's embarrassing. I was going for biology and they were like, no, you're gonna fit over here better because of your interests. And I was like, okay, so you're calling me a weirdo? Taking me from the one of the biggest STEM programs at the school to one of the smallest. I'll get off my high horse in a second. But I also have a minor in chemistry and statistics. So I do also think that makes me more credible to tell you about actual statistical significance in a study. Something about research that's unfortunate is a lot of the times you have to do trials on animals before you can get to human trials. It just has always been something that bothered me. I never had to directly work with rats, but in college I was using materials that were made from mice and rats, and they were like, do you want to go see them? Do you want to try and do like the extraction? Because we were using their spinal cords to study a specific protein. I said no, because I did not want to see poor rats suffering. It makes me really sad. It's time to actually get into some science. I will be reading out loud, and don't judge me. The purpose of this study that I've mentioned but haven't really fully gotten into is to test the effect of stress and music on depression and anxiety symptoms in mice. Depression is the most common psychiatric disability, and there's no known cure or mechanism for the disease. The ADA recognizes depression as a disability, and depression affects almost 25% of the world's population, which is ginormous. If there's 8 billion people in the world currently, that means that 2 billion people are affected by depression. Even though the stigma around mental health has gotten a lot better just in the last 5 to 10 years, depression is still affected by mental health stigma in society, and it's often brushed aside. This makes the situation 20 times worse. I know from experience, and it really helps just to have someone say, I hear you, I understand, and we will help you. Depression is treated most commonly with talk therapy and sometimes medicine as well. 1/3 of the population of people that have depression are resistant to drugs. To introduce a study, I'm going to talk about the study design. There were 4 groups of mice that went through the study: a control group with no changes in their environment, a group only subjected to chronic mild stress, a group only subjected to music, and finally a group that was subjected to both chronic mild stress and music. Stress is relative, so to model stress for the mice, they were subjected to restraint, tilted cage, wet bedding, food and water deprivation, a tail nip, cold water swimming, and light inversion, which just means that they were subjected to dark during the day and light at night. Since music is also relative, the scientists curated a playlist that encompasses a wide variety of music with different instruments, vocals, and styles. To mimic normal human stress conditions, the stress factors were delivered during the day and the music was played at night for 1.5 hours. Each group of mice went through 5 behavioral tests, then tissue was taken from them and analyzed for different biochemical factors. The behavioral tests included an open field test, sucrose preference test, tail suspension test, novelty suppression feeding, and an elevated plus maze. The purpose of the open field test was to measure the curiosity and movement of the mice by placing them in the middle of a box, then the time spent in the center of the box and the distance traveled were recorded. The sucrose preference test was to measure the preference of sucrose over tap water, because rodents are naturally inclined to prefer sweet foods. The tail suspension test was to measure, quote, behavioral despair, which is the scientific way to say hopelessness or helplessness. The mice were hung from their tail from the ceiling. The time that they were immobile or not struggling to escape was measured. Novelty suppression feeding is exactly what it sounds like. The mice were starved for 24 hours prior to the test, then they were placed in the corner of a box with food in the middle. The time it took for the mouse to move to the food in the middle of the box was recorded. A longer period of inactivity shows anxiety behavior. Lastly, the purpose of the elevated plus maze test was to measure anxiety levels and exploration ability, and is shaped like a plus sign, with open and closed spaces for the mice to explore. The number of entries into the open and closed spaces and the time spent in each arm were recorded. It's getting worse every second, and I do kind of love it. The results of the behavioral test do fit the hypothesis. Overall, the stress plus music mice and the control mice showed no significant depression or anxiety behaviors during these tests. I looked like a live-action Cat in the Hat. Ooh, he's scary too. Contrary to the control and the stress plus music mice, the only group that showed significant anxiety and depression behaviors during these behavioral tests is the stress group. Once the behavioral tests were completed, blood serum and brain tissue were extracted from the mice that were in the study. I knew it was going to be funny, for me to talk about actual serious science and also be doing my makeup like a rat. I couldn't prepare myself for this. I'm not taking comments about this one whisker at this time. Also, Remy is going to absolutely slay. I'm gonna give him a cute little eye look. Some background about the biochemistry of depression. Depression can alter the brain's defense against inflammation, and it alters the availability of inflammatory factors. These proteins are just made to affect inflammation, so they can either decrease or increase it. These specific relevant inflammatory factors that were found by previous other scientists were measured through RNA expression in these mice. I like to think of RNA as the transcribed version of our genes, which is our DNA. Once again, the stress plus music mice showed levels of different inflammatory factors similar to the control group, just like the behavioral tests. The stress mice showed similar patterns of abnormal expression of inflammatory factors that were measured. Have not been in the lab for about a year now. This was like one of the first scientific papers I read all the way through in the last few years. These results suggest that music plays a role in regulating inflammatory factors in the brain. The death and deterioration of brain cells or neurons in the hippocampal region is known to be abnormally high in depression patients. To test for neuronal decay, neurons were tagged with immunofluorescence, which is basically just a glow-in-the-dark tag on specific proteins. You can look at them under a specific microscope and you can see which neurons are lighting up. The stress-only mice showed the most significant neuronal death in their hippocampal region. Neither the control or the stress plus music groups showed any significant neuronal death in the hippocampal region, similar to someone without depression. Since neither of the groups of mice showed significant neuronal decay, this suggests that music may play a role in preventing apoptosis of neurons, which apoptosis is just a fancy word for cell death. The third neurological test that they performed was they were measuring corticosterone levels. You've probably heard of cortisol, the stress hormone, and to lower your cortisol, you need to eat a healthy diet, work out, blah, blah, blah. All of the things that medical professionals tell people with depression. Corticosterone is a hormone that's released in response to stress. Corticosterone can damage neurons by decreasing the brain-derived neurotrophic factor, which can just be abbreviated as BDNF. BDNF protects neurons and helps connections between them. BDNF levels on each subject was tested, and you guessed it, the stress-only mice showed the smallest number of BDNF, and the control and stress plus music mice showed similar normal levels of BDNF expression. The last test was measuring oxidative stress levels. Oxidative stress is linked to many conditions, including neurodegenerative diseases like ALS and Parkinson's, cardiovascular diseases like heart disease, type 1 and 2 diabetes, depression, of course, and many more that I did not mention. Oxidative stress is the buildup of reactive oxygen species, which are molecules that are produced by the body that are damaging to cells, proteins, and DNA. The body has these scavenger molecules to combat these reactive oxygen species that find them and turn them into molecules that are easily digestible by the body. I'm giving Remy a little cherry May heart because I feel like I shouldn't do any looks without it because I love it. Side note, this is actually what I studied, what I researched in college, and I actually won an International Youth Investigator Award for it. My biggest accomplishment in science, but I was studying ALS, or amyotrophic lateral sclerosis, and I was studying how the protein SOD works in the ALS mechanism. The researchers measured the level of reactive oxygen species in each mouse's brain tissue, and surprise to absolutely no one, the stress mice showed the highest level of reactive oxygen species, and the control and stress plus music mice showed similar results as a healthy normal person. All of these tests combined suggest that music plays a role in decreasing certain depression symptoms and can counteract certain stress responses from turning into chronic depression or anxiety. I actually had so much fun doing this video. I was a little nervous just because talking about science is a little nerve-wracking, especially when you're AFAB. People do not take you as seriously as you want them to. Just getting on here and being able to talk about my passion and also do another passion of mine that is kind of funny is really awesome, and I really appreciate you staying and watching. With that, thank you so much for watching. Here is my final little Remy look. If you liked the video, thank you so much. Like and subscribe. And if you want to see more of me in the meantime before my next video, you can follow me at Instagram and TikTok at Cherry May Drag.